Well, welcome to a special edition from Straight Shoot News, The Good Fight Forevermore. My name's Ed Bandana. I'll be hosting the show tonight. Tonight, we're going to bring to you the Rand Paul hearings. Everything that's been unfolding in the past 3 weeks have been out of desperation, and it all broke. Rand Paul finally got a Senate hearing on that, and all these legitimate questions came up that you're going to hear tonight. What he did is he put the deep state and all these people into a complete act of panic. That's why I think they did the raid. They've come forward. Everything else, they need to stop Trump, and they need to stop the Congress and Rand Paul and the rest of the senators who are actually legitimately doing their job from getting this information out. Because before we begin the hearing tonight, I'm going to begin with this statement. It's not meant for shock value. But when we unfold all this factual information in front of your eyes tonight, and you're going to start thinking, none of these things that are happening really make any sense as policies that are being implemented in front of our eyes. When it comes right down to the future of our country and even the free world, why would anybody even be doing what they're doing now? I'll tell you what it appears to be. It appears we're talking about what's taking place right now. It's looking more and more like treason. And treason carries possible death sentences for the major players in this conspiracy. We definitely have far too many people that are caught up in this. So we're all hoping for the November election, and you'll hear clips tonight with everybody, so we'll get him in November, we'll take over Congress. So I think his wife, Fauci, made that decision to retire at the end of the year before the Republicans actually take over Congress. Because if we do have the election and they do win, they'll be taking over probably, it'll be the second week into January. I think Fauci is going to make a run for it. But we'll see where that goes. By the end of tonight, I'm going to get through part 2. You should be, at the very least, if you don't believe what I'm telling you, you'll take what I'm saying into consideration. And we're going to start demanding immediately action needs to be taken. We're not talking about political parties here. We're talking about the future of the human race. I said, not trying to shock anybody, but just we'll listen to these segments and we'll talk about them as soon as we're done. Want to roll the opening one with the questions here? Part 1, roll it.
Do you think the USAID, whose main job is to provide humanitarian aid globally, has the oversight for programs and experiments like Stop Spillover and Deep VZN, which are not humanitarian in nature?
I think there's a very strong humanitarian case for preventing pandemics. I think that the absence of security oversight means that USAID was probably just not aware of the security consequences of their work. And it remains to be seen whether they will decide that it is inadvisable to maintain a ranked order list of the most threatening viruses.
So do you think they have the oversight ability to handle this job?
It's unclear exactly who they're seeking advice from. My understanding is that they are seeking advice from folks with greater security expertise. And the real question is what actions are going to come of that.
So would these programs go through a P3CO review?
My understanding is that federally funded research does go through P3CO review. However, it is unclear whether the basic Find the Pathogens program would go through such review, because until you find it and at least run some characterization to determine whether or not it looks like a pandemic virus, it would not necessarily be regulated. And as previously mentioned, due to the transparency issues with that committee, it's very much unclear what their remit is and is not.
Do you know who's on the panel for P3COs?
I do not.
Why wouldn't, is it not public?
My understanding is that it is not public.
Why wouldn't it be public?
That is an excellent question.
So Do any of the witnesses know why it wouldn't be public?
No.
Is it part of
I
the?
know it isn't public, and I don't know why it is not
So it's part of our federal government, right?
correct.
And so what, do they think Americans are not smart enough to understand it?
You'll have to ask the people at NIH.
Do you know how they made the decision not to make the names public?
No.
Okay. So for each of you, do you think that the P3CO review is comprehensive enough on NIH grants, or do you think gain of function grants have been approved without a P3CO review?
Let's go to Dr. Ebright's. I want to leave him out of the, and we'll go through each of you. Dr. Ebright, would you like to respond to that?
Yes. So, as I mentioned in my summary statement, there have been only 3 P3CO reviews in the 4.5 years that the P3CO framework has been in effect. The majority of gain-of-function research of concern, enhanced potential pandemic pathogen research supported by NIH has not undergone P3CO review. It has not undergone P3CO review for the simple reason that the NIH has not identified and flagged the proposals as subject to P3CO review and has not forwarded the proposals for P3CO review.
When we ask the other two to respond as well.
Yeah, I think just echoing Dr. Ebright, it's been a failure, I think, at this point in time, and so we need to find an alternative, which is perhaps to take it out of the NIH, make the oversight outside of the agency that's funding.
One major problem is that gain of function is a terrible term. It applies to most of biotechnology in the raw, and you can try to add qualifiers as you want. But it also inherently does not catch efforts to identify perfectly natural but nevertheless highly lethal pandemic-capable viruses. And it really doesn't matter where the thing comes from. What matters is, do you know that there is a good chance that it causes a pandemic? And again, maybe you don't think we can ever be confident more than, say, 50% for a given virus. But if you get a list of 8 viruses that you're 50% confident, it's possible to make all 8, let them go, and you have got pretty good odds there. So I am concerned by efforts to continue to focus on gain of function because it is so ill-defined, and it seems more productive to narrow in on the classes of experiments that can substantially increase our confidence that a virus is pandemic capable, wherever it comes from. And I certainly echo the calls for external security oversight.
Do you think there's appropriate oversight of existing research after it's been approved to ensure continuous compliance? Or do we
I would say there is not. Importantly, the P3CO framework does not mandate compliance. If the P3CO committee makes a decision that the research may not proceed, that decision is only advisory to the funding agency. It is not mandated for the funding agency. The funding agency is free to accept or not accept the decision and is free to determine whether to monitor or not to monitor the progress of the work. This is a major shortcoming.
Thank you.
I just want to interject on the definition, whether gain of function is a good definition or not. That began with the NIH. They gave us a definition and we started with that. And so I do think Dr. Esvelt's making some good points that we ought to be concerned with viruses that are not created, but they actually come from nature that could cause pandemics. So I think that's part of this discussion is to try to figure out where we get to. Senator Johnson.
Let's take a look at what just transpired there, and then we'll take some questions if you got them. Right now, this is a hearing now. Live. Finally, they got one. This is the first one. And the witnesses are Dr. Ebright, Dr. Esvelt, and Dr. Quay, all highly qualified professionals. So now it's Senator Scott from Florida, and they're talking about this PC30 review. This PC30 is supposed to review between the funding and all the research that's being done. Turns out, we just found out it's made up of a secret panel. Only 3 reviews have been conducted in 4.5 years since it came into existence. The NIH, headed by Fauci, has not forwarded the information for any PC30 reviews of the funding he sent to Wuhan. PC's committee, as Dr. Ebright said, is only an advisory committee has no power to prohibit any funding. So if the NIH, which is headed by Fauci, has not sent anything over there, these people are just running amok. I mean, we have this. This protects our rights and also tells the federal government they're in charge of national security. This is a national security risk. So with that, I'll take any questions if anybody's got any. If not, we can move on to the next one, which is Senator Johnson. Okay, with that, we'll move on to Senator Johnson's questions.
Hey, Mr. Chairman. How long have we had gain of function capabilities? Is that with the CRISPR technology?
Discussions have been underway since 2002 and 2003. The first examples involved reconstruction of previously eradicated or extinct pathogens. Those presented a prototype for understanding experiments that would create new health threats and the need to address them. And again, we're talking a 2-decade-long discussion.
But again, the technology emerged, or they started discussing and then developed the technology? The
discussions occurred as the technology emerged. It became possible to do this effectively starting at the beginning of the millennium. The technologies have increased in sophistication and have increased in ease and decreased in cost over time.
Yes, talk about the ease and the cost, because I've heard is very accessible now and it's very cheap, and you can basically, the knowledgeable individual can basically do this in their garage.
I think that's an exaggeration, but as Dr. Esvelt has pointed out, given the genome sequence of a virus, it is typically possible to reconstruct infectious particles of the virus, and to do so for costs well under $10,000 US in 1 person month or 2 person months. So for an equipped laboratory, the kind of laboratory that would be present in any state program, and that is present in many research laboratories at academic institutions, this is eminently possible.
So again, reconstructing a virus is one thing, but my understanding what the at least theory might be with SARS-CoV-2 is there's gene splicing that occurred here, and some very unusual markers in this furin cleavage site, and it would beyond my comprehension exactly what that means, but talk to me a little bit about the whole gene splicing aspect of this.
So there's 2 ways to edit a virus. Nowadays, the easiest way is usually to assemble it from scratch using synthetic DNA. But if it's large, then in some cases, it's better to create the altered piece that you wish to insert into the virus and then use a tool such as CRISPR to do the insertion into the backbone. With respect to the cost, the first virus with a chemically synthesized genome from synthetic DNA was made in 2002. Since then, the cost of gene synthesis has fallen by roughly 1,000-fold. So today, the cost of ordering the components of an infectious influenza virus, for example, the synthetic DNA costs less than $1,000. And that does not require any further editing. That just requires following the reverse genetics protocol, transfecting it into the cells to get the infectious virus. And I estimate that there are around 30,000 people who can do that who have doctorates. And you can say 125 virology PhDs per year in the United States. That's roughly 1/3 in the world. There's probably 4 times as many people who have degrees in other disciplines, such as mine, who can do it. Assume a 20-year career, and that's 30,000 people, add a few technicians.
So
Was there a specific incidence or something that concerned people that caused the pause?
yes, there was experiments in influenza in the Netherlands and Wisconsin that took a virus that was 90% lethal but not airborne and made it airborne through passage in the laboratory.
And that occurred when?
2013, 2012.
And that caught the attention of who? I mean, who was alarmed by that and instituted the pause? I know it occurred under President Obama, but which member of our health agencies that,
Yeah, I think Dr. Ebright would be the best to answer that.
Dr. Ebright.
So the proximal impetus for the pause was a series of events, laboratory accidents at federal laboratories that have access to and storage of potential pandemic pathogens. The accidents included an anthrax incident at the CDC, another anthrax incident at a U.S. Army facility at Dugway in Utah, and the finding of unsecured vials labeled smallpox virus in an FDA NIH freezer in Maryland. And those three incidents occurring in close succession resulted in a hearing in the House Energy and Commerce Committee, and then action by the Office of Science, Technology, and Policy. So the pause was driven ultimately from the White House, from the Obama Office of Science, Technology, and Policy.
So listening to your testimony, I'm assuming all three of you would agree with this statement that this research, and I would say even the mining of dangerous potential pathogens, go crawl in a bat cave to try and pull these things out and bring them into a lab. There's certainly no benefit that overrides the risk. We shouldn't be doing this at all.
Yeah, I call it gain of function and gain of opportunity where you bring a virus back. And as I said, my analysis is that it hasn't contributed to the response to this pandemic.
So we shouldn't do it. I mean, we shouldn't be, we can talk about controls, but bottom line, we shouldn't have controls because we shouldn't even do it. Is that your position as well?
For balancing the potential benefits of prevention against the risk of accidents, it can go either way depending on the numbers you use for those. You can reasonably come out with either answer. When you add the misuse case, that is what absolutely blows it out of the water.
Dr. Ebright?
I believe a strong case can be made, or a case can be made, that certain components of gain-of-function research of concern, particularly components involving pathogens that are currently in human populations, are categorically separate and more justifiable than other components of gain-of-function research of concern. For example, currently SARS-CoV-2, the virus responsible for COVID, is present in millions of humans and is generating variant after variant. Gain-of-function research of concern on SARS-CoV-2 involving the creation of new variants and analysis of the threat posed by them arguably can be justified because this is not creating new health threats that won't arise without intervention, but is addressing a health threat that's in place currently. For that reason, and for reasons like that, I believe enhancing the oversight of the research is a more effective and more prudent strategy than simply banning it.
I would say improve oversight, but would you also agree dramatically limit it?
Absolutely.
Okay.
Okay. Let's unwind that for you. So basically what we just heard from Senator Johnson was questions about how this CRISPR technology was developed in 2002, 2003. I believe it was Dr. Esvelt who explained the ease of the cost of under $10,000 for CRISPR technology that can actually mutate DNA and viruses to mesh. And now it's under $1,000. That's quite shocking in some ways. And he was talking about how they already did this, 2002, 2003, when they started this. If you think about it, and I do have a timeline that we'll post a link to on a rough draft of a book that I'm writing now of all the viruses, everything that's been created since 2002 and 2003, since this CRISPR technologies come into play. So what we have is just prior to this CRISPR technology, just for those of you who don't know already, Dr. Fauci, Bill Gates, and Klaus Schwab of the World Economic Forum began a foundation. I call it the Unholy Alliance. Ever since then, a lot of diseases have erupted. viruses that come out. And what's said here too is talking about how they already took a flu virus and made it airborne in 2012, 2013. And that's when there were accidents in the lab. What they're playing with here is pretty dangerous. You know, and they're calling it a gain of function and gain of opportunity. The limit is absolute. And they actually, they tend to lean towards how this could be stopped. I don't see how it could be stopped if this technology is so inexpensive and so available. I mean, we have a problem here. This is where we're starting to look at the crimes against humanity part of this. And I think we just lived through the nightmare of what it can do to the human race. Well, let's move on to Senator Marshall here. And he's got some even more concerns to add to this. Go ahead. Go ahead.
I think it's important to not only identify the true problem, but also talk about where we've been. And you all can help us fill into some of the pieces here when we talk about gain of function research. It was late in 2011 when the NSABB, which is the NIH's advisory board, stopped 2 scientists from publishing an influenza gain of function study that I believe Dr. Ebright was referring to. And they stopped it because they were afraid it could educate bioterrorists. So this is 2011. Over a decade ago, scientists had figured out how to make H5N1, which is highly pathogenic avian influenza, more contagious. In 2012, those 2 scientists and 39 others implemented a voluntary gain of function research pause on influenza experiments. In early 2012, Dr. Fauci encouraged all influenza scientists to pause gain of function and said, and I'm quoting Dr. Fauci, 2012, "It's essential we respect the concern of the public domestically or globally and not ask them to take the word of the influenza scientists." It's interesting to me that Dr. Fauci was focused on the messaging, but he still wanted to continue the gain of function research. Again in 2012, Dr. Fauci also said, almost prophetically, that he worried about unregulated laboratories, perhaps outside of the United States, doing work sloppily and leading to an inadvertent pandemic. And he went on to say the accidental release is what the world is really worried about. I go forward to 2014 now, after biosecurity accidents in the United States research labs, which our witnesses have talked about, the Obama White House implemented the second gain of function moratorium on influenza plus MERS and SARS because of the potential risk of lab accidents and inherent gain of function danger. But gain of function still continued at the University of North Carolina. Research later that we shared with Dr. Shi, the bat lady. Nevertheless, clearly the U.S. government and Dr. Fauci knew that the viral gain of function research was very concerning. And almost counterintuitively, while Dr. Fauci encouraged United States scientists to pause their GOF studies, Dr. Fauci offshored the pause research to China, not once, but twice. In 2012, Dr. Fauci gave a new grant to Peter Daszak's EcoHealth Alliance for influenza research in China. And then again in 2014, Dr. Fauci gave another grant to Daszak for SARS research in China. Daszak partnered with who? The Wuhan Institute of Virology. In late 2017, the NIH announced a lift on the gain of function moratorium, what became known as the P3CO framework, which we referred to, apparently without consultation from a Senate-confirmed State Department head or national security leadership. Also significant, there was no OSTP director in place and only an acting HHS secretary at the helm. So what was the result of this? NIH essentially lifts the moratorium on their own by slipping it in between administrations and self-policing. And today, we can't see the research record for Dr. Fauci's offshore projects because the Chinese Communist Party supposedly has EcoHealth records and NIH resists sharing theirs. So I'll get to my question now. Dr. Ebright, Could EcoHealth's research in China have led to the COVID-19 pandemic and Dr. Fauci's worst fears that a lab accident in a foreign lab became reality?
Yes. Lapses in U.S. oversight of gain-of-function research of concern may have caused the current pandemic and could cause future pandemics. The U.S. government funded high-risk gain-of-function research and high-risk enhanced potential pandemic pathogens research at the Wuhan Institute of Virology in 2016 to 2019. The research overlapped the pause that was in effect in 2014 to 2017 and met the criteria to be paused, but was not paused. The research also overlapped the subsequent policy, the P3CO framework that has been in effect from 2018 to the present, and met the criteria for federal risk-benefit review under the P3CO framework, but did not undergo federal risk-benefit review under the P3CO framework.
Thank you so much. I have to stop and point out, too, that U.S.A.I.D., who is knee-deep in this type of research, is part of the State Department where they can get the security advice that they should have asked for before they cleared this with P3CO. Certainly, I believe that this virus came from Wuhan, China, and that it is a product of gain-of-function research. This is a bipartisan national security issue, like several of our witnesses have testified, that this viral gain-of-function could become, and has become, a weapon of mass destruction. And all that, this became a nuclear hand grenade is what happened. Dr. Quay, then Dr. Esvelt, considering the extreme risk of this research and the incredulous obstruction by the NIH, USAID, EcoHealth in China, should Congress immediately pause this dangerous research?
I think that's an appropriate step for Congress to take.
Okay. Dr. Esvelt?
I think it would be somewhat dangerous to attempt to pause gain-of-function research when it's evident that that term is so malleable as to be evaded at will, and also could plausibly do damage by applying to science that is not specifically directed at potential pandemic pathogens.
Are there any countries that you would say we shouldn't be doing this type of research with?
When it comes to identifying pandemic-capable viruses that could kill millions of people and will necessarily be shared with scientists worldwide who will be able to access them, I do not think that we should be doing it. I do not think that China should be doing it. I do not think that anyone should be doing it, because it is expected to kill 100 times as many people as it might save, even if we could perfectly prevent an identified natural virus from spilling over.
Whoa, that's a lot to unfold there, but boy, did he get the truth out. Let's see. They were concerned about creating bioterrorists. Well, of course, with that kind of technology available and the reasonable price, you could create quite a few bioterrorists. Fauci was even concerned in 2013. 2014, the White House was concerned. That was under Obama. The University of North Carolina lab incident. This is all justifying what many of us were saying back in 2020. We knew about this. There was an internet blogger who's does intense research on this. He released it. He's been banned almost everywhere that he comes on right now. We may see if we can get some clips up and put it up on maybe Rumble and BitChute or Brighteon. We won't mention his name here, so this doesn't get knocked off. But then they're talking about how Dr. Fauci shared all the research from the labs in the U.S. with the bat lady who was collecting the virus in China at the time. And the research continued in Communist China, who is our enemy, because it was barred in the U.S. We'll probably hear more on EcoHealth Alliance of Peter Danzig and funding, and we're going to have that in round 2 and part 2 of the show tonight. And that should surprise you, or not. So Communist China refuses to share any information on the matter. Matter of fact, you'll see, and I think it's in part 2, how they took down the website that used to share the information with the other scientists. and I think it was April of 2019. That was right around the same time, because it was May of 2019 when Communist China, it was published in the, I think, Chinese, South China morning paper. That's when the Communist People's Party of China declared war on the U.S. Many people don't know about that, but it's a fact. They did. And Communist China refused to share up. The research was not paused. It did not go through any review at all. They keep making that, although qualified by the rules that they set up to go by to that. That's why we needed this congressional review, which is their duty under this, the Constitution. The U.S.A. did nothing. So, as it was described, the weapon, this is a weapon of mass destruction. They called it a nuclear hand grenade. It's killing hundreds of times more people than it could ever save. And they told us right there, we should not be doing this. So why are they doing it? But now we're going to move on to Senator Hawley. He looks at this more from a bioterrorist and weaponry and a military weapon. Let's listen to what he's got to say.
Senator Hawley.
Thank you, Mr. Chairman. Thanks to the witnesses for being here. Dr. Quay, if I could just start with you. You said in your written testimony that the genome of COVID has some of the hallmarks of gain-of-function research, and in particular, 3 genomic regions you say have the signature of synthetic biology. One region has features of the 2 types of forbidden gain-of-function research that are associated with bioweapons development. And you said in your opening remarks that you believe COVID-19 was the product of gain-of-function research and was from a lab leak from the Wuhan Institute of Virology. My question, I guess, is, do you think that China engaged in a cover-up to prevent the world from knowing the true origins of this virus in a lab leak?
I think there's abundant evidence that they have not shared all the information they had at the time. They continue to not share information. I could give you a laundry list of 20 things that they've done, starting with a website with 21,000 viruses on September 12th. 2 a.m., someone was in the Wuhan Institute of Virology. That had been available to virologists for a decade. It was taken offline. It's not been returned. We've asked to see it. No one that I know of has ever seen it. It goes on from there.
Are you concerned with the continuation and expansion of Chinese gain-of-function research?
Well, I think I testified here that in December 2019, they were doing synthetic biology on a cloning vector of the Nipah virus, which is 60% lethal. We just experienced a 1% lethal virus. My estimates would be that could set us back a millennium. The Black Plague was a 20% lethal event, and it was 250 years for civilization to return.
Let me ask you this. How safe were the testing conditions at Wuhan, to your knowledge?
Well, I think that a lot of the Western virologists actually used the findings of that as a way to get around saying it was okay at the beginning. All of the work that I've described is being done at what's called BLSA 2-3 level, which is commonly spoken of as a dentist laboratory level of biosafety. So maybe a little higher than that, but that's not a bad euphemism.
You said, I think in your testimony, this is the most dangerous research that you have ever encountered. What makes this particular research so dangerous?
If you're doing experiments with a pathogen that is 60% lethal but is not airborne, and you make it airborne in the laboratory, and someone walks out with it. Nipah has a 21-day incubation period. It's perfect for widespread spread without being detected. We couldn't afford we can't afford 10% lethality.
Yeah. Dr. Ebright, let me ask you about the merits of gain-of-function research, because I was struck by something you said in your written testimony. You said gain-of-function research has no civilian practical applications. From a research perspective, then, why do it? I mean, what's the value, the real value of gain-of-function research?
Not a matter of value, but incentives, particularly incentives within the academic research ecosystem. Gain-of-function research of concern is fast and easy, much faster and much easier than vaccine or drug development. And gain-of-function research is publishable, and gain-of-function research is fundable. With those 4 incentives in place, fast, easy, fundable, and publishable, the research will be performed.
What is,
Eliminate any one of those incentives, and it will not be.
so thinking about China for a second, what's China's interest in gain of function research?
They have witnessed the United States leading the way with gain of function research. Most gain of function research of concern performed to date has been performed either in the U.S. with U.S. funding or overseas with U.S. funding. China has wished to be part of that and has participated in gain of function research of concern in China with U.S. funding, and has also supported gain of function research of concern in China entirely through Chinese programs.
So let me ask you this: gain of function research and bioweapons. What's the connection there? I mean, what role does gain of function research play?
As I mentioned, there are no civilian practical applications. There are immense bioweapons practical applications. As you've heard from Dr. Esvelt, the potential pandemic pathogens that can emerge from such studies are potential weapons of mass destruction, inexpensive, accessible, easily distributed weapons of mass destruction.
Let me ask you about some of the things that you have commented on with regard to what NIH and Dr. Fauci have said, and frankly, the lies they've been caught in regarding the coronavirus. I want to highlight two of them. In response to a congressional inquiry from October of 2021, just last year, the NIH attempted to walk back assertions by NIH Director Collins and Fauci that NIH had not funded gain-of-function research in Wuhan. You commented at the time saying, and I'm going to quote you now, NIH, specifically Collins, Fauci, and Tabak, lied to Congress, lied to the press, and lied to the public knowingly, willfully, brazenly. On May the 11th, Dr. Fauci said the NIH and NIAID categorically has not funded gain-of-function research to be conducted in the Wuhan Institute of Virology. You commented on that, saying the documents make it clear that assertions by the NIH director, Francis Collins, and Fauci, that the NIH did not support gain-of-function research are untruthful. So just expand on that if you would. I mean, what are the implications of Dr. Fauci's continued blatant dishonesty regarding NIH's funding of gain-of-function research in Wuhan?
I stand by my statement. The statements made on repeated occasions to the public, to the press, and to policymakers by the NIAID director, Dr. Fauci, have been untruthful. I do not understand why those statements are being made, because they are demonstrably false.
Can I ask you just about, in my few remaining seconds here, let me ask you about an effort to shut down any kind of questioning of the origins of COVID. On February 19, 2020, a group of virologists and others published that famous letter, infamous letter in The Lancet, which said, among other things, "We stand together to strongly condemn conspiracy theories suggesting COVID-19 does not have a natural origin." And of course, we later found out that the Lancet letter had been organized by Peter Daszak, president of EcoHealth Alliance, which we've discussed today, operated a lab in Wuhan with a $600,000 5-year annual grant of taxpayer dollars from Fauci's NIAID to study bat coronaviruses. That letter conveniently concluded by stating, "We declare no competing interests." Many people designate this letter as the first effort to quash any kind of debate about the origins of COVID-19. Do you think that labeling the lab leak theory as a conspiracy theory so early on had the effect of slowing down investigations into the origins of the virus?
It certainly had that effect, but the Lancet letter that you described was only one of two efforts to impose the false narrative that science shows SARS-CoV-2 entered humans through natural spillover, and that is the consensus view of scientists. One of the efforts was the Lancet letter you discussed. The other effort was coordinated and orchestrated through the National Institutes of Health, through the NIAID director, Dr. Fauci, and the NIH director, Dr. Collins, and resulted in the publication of an opinion article entitled "Proximal Origins of SARS-CoV-2," making the case, again, that SARS-CoV-2 could not have been a product of research-related spillover.
Thank you very much. Thank you, Mr. Chairman.
Well, That's pretty alarming. When you look at it from a weapons point of view, it has the signature of bioterrorist weaponry. And once again, China will not share any information. They took down their website. They referenced the Nipah virus. And they tell you it's 60% deadly. Think about that. They compared it to the Black Plague, which is 20% deadly. And it took 250 years for the human race to recover from the effects of it. I mean, these are alarming statements. The gain of function research has no practical civilian benefits, which means this was developed as a weapon. Now, if it's developed as a weapon, why would the U.S. be sharing it with our enemies in communist China? That's something to do some serious things. Here's what we're talking about, not just crimes against humanity. We're talking about treason. We are talking about treason. The incentives is fame and fortune. That'd probably be the least of the evils. Was China concerned with what the U.S. was doing with the research? Sure. Same thing, the U.S. should be concerned about what China was doing with the research or is doing. Warfare weapons of mass destructions. Collins and Fauci lied about funding the research. And they used the same old playbook. They covered up, calling it all a conspiracy theory. Peter Danzig, Eco Health Alliance, had a letter published in the Lancet, in the medical magazine that a lot of people read that are into the medical industrial complex, as we call it here. You know, and that letter denying all this cost millions of lives, slowing down the reaction to the virus. NIH, Fauci, and College published a second letter stating it didn't come from the lab. So can you see why we opened up with making the point about how Fauci's retiring? Maybe he doesn't feel comfortable. Maybe he thought he was comfortable and thought he had some kind of shield with the Democrats in charge. Because we know they leveraged this to make a mess out of the 2020 election. And now he doesn't. So is he going to actually testify in front of Congress? Or is he going to make a run for it? We'll see what Senator Kennedy from Louisiana has to say about that in part 3. But now let's hear from Rand Paul. Let's listen to him put this all together.
Thank you. Had there not been a pandemic, I think there would still be a need for this hearing. This discussion, Dr. Ebright had gotten this started back as early as 2003, 2004. Others have commented on the danger of being able to manipulate influenza viruses to be used as either weapons or by accidental release. But I think given that there was this pandemic, that 1 million Americans died, I lost friends. I have good friends to the pandemic. I think we should be curious. I just am perplexed by the lack of curiosity on some as to know, are there any precautions we can take? Are there any kind of government oversight that we could do to try to prevent this from happening? Now, some will say, well, we can't prove it came from a lab. That's in all possibility true, that we can't prove it, but there are arguments to be made and examination of facts to give us an idea of whether it might have come from a lab. And even if we didn't, I think that this could have come from a person in a lab handling a virus, if it was a virus out of nature, and we've discussed that as well. But I do think that we have to get to the truth of the matter of whether or not dangerous research was going on that should have been reviewable. We had a pause of gain-of-function research, but then we had research occurring during the pause that should have gone to this committee, this P3CO committee, and didn't get to the committee. And I think Dr. Ebright's described it well. he says that in Wuhan in 2016, 2018 period, they were constructing novel chimeric SARS-related coronaviruses that combined the spike gene of bat SARS coronavirus with the rest of the genetic information of a SARS-1 related virus, one that was already known to have lethality. And they found that it could efficiently infect human airway cells and exhibited up to a 10,000-fold increase in viral growth. But then when we've asked before, is this gain of function, we get sort of arguments and protestations that this isn't gain of function, as if this is no big deal and the experts looked at this. But as we look farther into this, we find that the experts never looked at this, that it's sort of a select-in kind of program to this committee. It doesn't go looking for dangerous research. It looks at it if you come to them and say, hey, I think I've got gain-of-function research. Do y'all want to look at my research? And so this opting-in aspect of this, but I think it's important that we get to the truth. You know, was there research going on in Wuhan that was dangerous? Was it funded by the NIH? And should it have gone through this committee process? I think by the definition that they have given us, you know, gain of function, I think I agree with Dr. Esvelt, could be better defined. And particularly if we're going to have oversight on this, we're going to have to figure out what our oversight's going to be. So by all means, moving forward, we need to ask and include the scientists to get a precise definition of what we're talking about if we want to have more oversight. But we have to look back before we can look forward, not so much to assign blame, but to figure out, is it really necessary? Do we need to be having hearings on this? Should we have follow-up hearings? Should we have legislation? And if 1 million people died and there's a chance this came from a lab, I think without question we should. Both sides of the aisle should be looking at this. So I guess my question, I think it's pretty clear, but I'd like to go through everybody, even though Dr. Ebright has said this was gain of function, to each of the three witnesses. Was the research where you take the backbone of a SARS-1 virus that has known lethality and you mix it together with an unknown bat virus S protein genes to create a new virus, was this gain of function according to the NIH definition? And should it have been reviewed and discussed by this committee that was supposed to prevent dangerous research from going on? We'll start with Dr. Ebright.
So, as you mentioned, the Wuhan Institute of Virology constructed novel chimeric SARS-related coronaviruses that combine the spike gene of one coronavirus with the genetic information of another. They showed that the resulting viruses efficiently infected human airway cells and efficiently replicated in human airway cells. And they showed that the resulting viruses exhibited up to 10,000-fold enhancement of viral growth in lungs and up to 4-fold enhancement of lethality in mice engineered to display human receptors on airway cells. Based on those facts, and they are indeed facts, the research was gain-of-function research of concern subject to the pause and was enhanced potential pandemic pathogen research subject to the P3CO framework. Nevertheless, due to the failure of the NIH to forward the proposals for review, the work was not paused and there was no P3CO review.
Dr. Quay.
So the Wuhan Institute of Virology is unique in the entire world. Before 2019, 65% of all publications on coronaviruses came from that single institution. They're unique for two reasons. For almost a decade, they were going into bat caves throughout China and actually back into Africa as well, 20 visits a year, and bringing these samples back to the laboratory. So on the one hand, they had the largest collection of raw material backbones from nature to then do gain-of-function research on. They trained in Galveston, Texas, and in North Carolina, and were doing experiments, published experiments between 2015 and 2019. And I believe it's the confluence of those two activities, gain of opportunity, bringing things back from bat caves, and gain-of-function research that led to the pandemic.
Dr. Esvelt.
On the list of experiments that you would need to perform in order to learn whether a novel virus could potentially cause a pandemic, you would need to test growth in human primary cells, such as human airway epithelial cells, and you would need to test transmission in a suitable animal model. So the question is, if they were not intending to determine whether a novel recombinant event between these coronaviruses could lead to something that might kill millions of people, then why were they doing it? If there was no chance that it would come up with a result that looked like it was more dangerous, what's the point? What's the scientific hypothesis? So again, whatever you call it, What they were trying to do was identify a biological agent that has a good chance of being able to kill millions of people if released. And they shared the description of what they did, and they shared the genome sequence because they thought that this would make us safer, because they think that knowing which viruses in nature might cause pandemics makes us safer. They did not consider the security risks. And it's worth noting that both USAID and NIH funded those particular coronavirus chimera studies. USAID, to my understanding, has since disavowed those chimeric recombination studies and announced that they will only focus on finding natural pandemic-capable viruses, which is at least a step in the right direction. But again, I would call that gain of function. Another reasonable scientist would say, no, that's not gain of function because the term is so ill-defined.
Even beyond the term, though, would it be qualified as dangerous research that actually should have gone before this committee, the P3CO committee, and been reviewed?
Well, here is where you come back to the problem of thinking this as a health and safety issue rather than a national security issue. The question is, why are we trying to identify readily accessible agents that could plausibly be used to kill millions and will, as soon as identified, fall into the hands of all of our adversaries, as well as perhaps individual terrorists who would just want to use them? The fundamental principle behind even wanting to do these experiments in the first place is, I think, a fundamental threat to not just national security, but international security. It's just hard to see why you would ever want to do this when you think about the misuse potential. And I haven't seen anyone else publish a numerical model of that.
Concerned yet? Are you getting the point? Well, let me make a note right now, just so before I lead into any more about this. I think about a year and a half ago, I reviewed a forensic study, an official forensic study, that stated that none of these viruses has ever jumped from mammal species to the other mammal species. And and this dates back, these studies through forensic science, which is legitimate. I know I took a intensive master's degree course on it in college. So I can safely say that when they do these studies, they have this proof. And we presented it to you. And they were actually looking for more funding to go back and look at it even deeper. But for thousands and thousands of years, none of these viruses has just jumped from species to species, like some of people are claiming now. So let's look at that, what we just heard here. Dr. Ebright, fact was, this was gain of function research. And another fact he said was, it was not paused. Even when they were told to be paused by, and this was under President Obama and under Trump. What is going on here? Dr. Quay, it came from a lab in North Carolina. Like I said, we found this out back when the epidemic first broke out before the lockdowns. And in Wuhan, Dr. Esvelt. This stuff needs to be tested in an animal, or let's change that word from animals to mammals, like we just said to you. Or it's never jumped from mammal to mammal. So who are they testing this on? Are they testing on us, the human race? Only now is the U.S. NIAID going to limit studies and experiments to only what's created naturally, like Dr. Esvelt said. And he also pointed out what is being done is being mistaken as a health and safety issue when it's actually a national and international security risk. Let that sink in. And now you see why we're saying, run, Fauci, run. And his accomplices have all got a lot to be concerned about. Because congressional oversight, which Congress has under this, for many reasons, as far as between funding, national security, this is what is going to be the demise. But who's going to enforce it? That's our concern. So let's run this next little piece through Rand's question and then roll this one out.
People have said, well, this, the closest relative that we found is only 96% identical to COVID-19. This couldn't have come from the lab. They've also mistakenly accused those who say it came from the lab of saying, oh, it came from this particular variant. But I think what people who are saying that this could have come from a lab are saying that there could also be possibly other viruses that are closer that were manipulated, or that the one that is 96% analogous to COVID-19 could have gone through serial cell culture and become COVID-19. I guess I'd like to ask each three of you whether or not the variant that is 96% analogous to COVID-19, could it through serial passage be transformed to COVID-19? Is it possible? Is it so far away that you can't do it experimentally? Could you do it through gene splicing? Could it be done? Or is it something that argues that this couldn't have come from the lab? We'll start with Dr. Ebright.
The closest relatives are more on the order of 97% identical to SARS-CoV-2 in genome than 96%. Viruses with that level of genetic difference cannot rapidly, in the time scale of weeks or months, move from their state into being a proximal progenitor of SARS-CoV-2. However, in the laboratory, those viruses can be combined at will. They can be combined in particular using a method that would be described as constructing a consensus genome virus. To construct a consensus genome virus, one takes the sequences of several related viruses, identifies the most commonly observed nucleotides at each position in these sequences, and then synthesizes the nucleic acid corresponding to the average, if you will, the consensus genome for the group of viruses. This has been done successfully in coronaviruses. This has been done and published a decade ago in coronaviruses. That kind of research could have been done using viruses that are on the order of 96 to 97% identical in their genome sequences to SARS-CoV-2, and with 2 or 3 or more such viruses, genome sequences, one could develop a consensus. And that's just one of a series of potential routes by which one of the known viruses with 96 to 97% identity could, through a laboratory, in a relatively short time, be transformed into a progenitor of SARS-CoV-2.
Dr. Quay?
The three sets of viruses that are closest to SARS-2 are one from southern China, RITG13, and a series of Banal from northern Laos. And as indicated, they're probably 1,200 letters different in the whole 30,000 letter alphabet. In nature, that takes approximately 40 years, so the most common ancestor is about 40 years ago, but most of that can be done in a couple of days in a laboratory. However, I don't believe we currently have the starting material, the backbone on which SARS-2 was found. I think it's one of the other 21,000 viruses in the database that was taken down at 2 a.m. September 12th, 2019.
So a great deal of information was destroyed by the Chinese.
It was taken offline, not available. I don't know if it was destroyed.
Right.
But yeah.
Dr. Esvelt?
If a PhD student proposed to take a 30,000 base paired viral genome and attempt to passage it in the laboratory to acquire 1,000 or so mutations, I would say that is not a PhD project. Go do something else. So I concur with Dr. Ebright that the only way that you could get something so divergent would be to computationally design it and synthesize it, which could certainly have been done. From what data set, and again, why? Why would you do such a thing unless you want to know what the ancestral virus was like and whether the ancestral virus was dangerous. There are basic science reasons why you might want to just know where they all came from. But at the end of the day, the reason why this research is of interest to us is the risk of pandemics. And so again, why would you run the tests to determine whether something was pandemic capable? And they certainly ran those on all of the other coronaviruses that they found and thought might be dangerous. So on the other hand, they never published anything like that, right? And presumably they would have. They published their data on the other stuff. So again, this is why I don't think we have enough information to know. But it was definitely not passaged in a lab from something that was 97%.
I agree. And one of the things that tips us off that they may have been trying was in 2018, they asked for money from DARPA. And in that money, they wanted to insert the furin cleavage site, which makes it highly infectious in humans. And so if they had the idea of that, they're asking for money, they must have thought, wow, we can do this and this is going to be a great experiment. Even our government finally at that point decided not to fund that, but what they're asking for, and this is why I think there was a holy cow moment when all of a sudden these scientists see the sequence of COVID-19, they're going, oh my goodness, didn't they ask us in 2018 to put that furin cleavage site in?
All right, let's take that apart. Dr. Ebright, he began. Again, naturally, these viruses cannot spread this rapidly. Dr. Quay, naturally, something like this would take 40 years, but can be done in the lab in days. Dr. Esvelt, it's designed. Why? Why would you possibly do it? Let's go back to Dr. Quay, September 12th, 2019. The info was taken down from the Wuhan lab site. Interesting date, considering how everything else lines up with it. Senator Paul, they asked for money for DARPA for funding, and they didn't get it. For those of you who don't know what DARPA is the Defense Advanced Research Projects Agency. So see, is recognized as a national security risk. And these people kept doing it. They didn't stop. Let's move on to this last bit here with Dr. Ryan to Dr. Quay when he asked some questions about the progressions of mutations and literal.
Point decided not to fund that, but what they're asking for, and this is why I think there was a holy cow moment when all of a sudden these scientists see the sequence of COVID-19, they're going, oh my goodness, didn't they ask us in 2018 to put that furin cleavage site in? And lo and behold, it's there. So what I'd like to ask, and I'm going to finish with this and then we'll have another round if some people would like to ask some other questions, is Dr. Quay, could you sort of lay out in as simple a fashion as possible 2 or 3 items about the virus that makes you think it came from, and I don't think anybody knows with 100% whether this came from a lab or whether it came from animals, but there is some compelling evidence that suggests it could have come from the lab. And, you know, even if it was a 10% chance it came from a lab, it's another reason for us to be concerned about having oversight on this kind of research. But can you give me 2 or 3 things that this virus has that make you think it's lab versus some of the evidence for MERS and SARS that it came from animals?
Yeah, there are 3 regions: the receptor binding domain, the furin cleavage site, and this protein 8th from the left called ORF8. With respect to the receptor binding domain, if you look at what happened with SARS-1, we have the virus sequence when it first was in civet cats in the markets, jumped into a few humans. We have the virus sequence then. It started infecting more. And then we have the virus sequence when a human-to-human passage could occur and an epidemic occurred. And so you can see the progression of mutations as the virus adapted from being in civet cats and then being in humans. The first jump into humans, it had only 15% of the mutations it needed to support an epidemic. Okay, let's flip to SARS-CoV-2. When you look at the virus that first entered the human population, out of all of the changes in the receptor binding domain, there are 200 amino acids, 4,000 possible changes. There were only 17 mutations that could make it a better virus. Its receptor binding optimization was 99.5%. And in fact, one of the 17 ended up being the Delta variant. So that kind of optimization, juxtaposed by the fact that there were no patients in Wuhan, 36,000 blood bag specimens tested for antibodies in 2019. Not a single patient was infected. Let's go back to SARS-1. 20% of all people in the markets were infected while the virus was practicing to set up an epidemic, 1% of the general population. So we would have expected 360 in the general population. In Wuhan, we had 0. Furin cleavage site has obviously never occurred in this related viruses, sarbecoviruses, that split from their cousins, the MERS viruses, around the time of William crossing the channel, 10,060. That was when the sarbecoviruses came. So there's never been a furin cleavage site, and the genetic sequence of it uses a code that's never been used, the CGG dimer as it's called, which has never been used before. Finally, ORF8, this protein that goes into the bloodstream and suppresses interferon response, so you're asymptomatic, and suppresses AHC antigen presentation, so you can't make good antibodies. This was the subject of 2 master's theses at the Wuhan Institute of Virology. I have found no Western scientists that worked on this location in the genome before 2019. And the protein is not present in MERS. It has a 5% homology in SARS-1. Between SARS-1 and SARS-2, there's a protein there, but it's only 5% homologous. But this master's thesis, the first one, optimized its function in suppressing interferon, symptoms of fever and chills, and suppressed its antigen presentation. The second one was making synthetic biology tools so you could move it around inside genomes.
So to reiterate, there's been no animals found that have COVID-19. When they did find that animals had the first SARS and MERS, they found it out within months. When they tested the animals in question, 90% of the animals had the SARS virus. So we haven't found any animals yet with COVID-19. And then most viruses that come from animals first aren't very infectious at first, and they infect a few humans. So you don't have a pandemic that does this. It smolders and then does this. During the smoldering phase, you find background antibodies that people have had it, even if they don't know they had it. So when they tested the background of people who were working with the animals that had COVID, they found 20% of them had antibodies to having had SARS.
SARS-1, yes, correct.
But then if we test the people in the marketplace, we're not finding that. If we look at the people in the Wuhan marketplace, we're not finding significant numbers that were positive, and finding almost nobody positive from the previous year that had been ill.
No, it's 0 out of 36,000.
Thank you.
Okay, let's roll this segment up with these thoughts, but let's take apart that piece. The progression of mutations exposes how they tried to make it a better virus. What sick bastards would try to create a better virus to infect the human race? And we'll go back to what Dr. Esvelt says. Why? This suppresses the interferon, which allows the body to make antibodies, which is what fights off disease in the bodies. Viruses that come from animals don't immediately spike. Like Corinne said, they smolder a period of time, which allows for those infected to develop antibodies. So it sounds like all the time that Fauci, his communist Chinese allies, that were doing this type of research, they were covering up. And what's happened in the meantime? How many millions of lives were lost? This qualifies as crimes against humanity, because they allowed this thing to spread worldwide from not being upfront, and at least have a reaction from the medical community, the legitimate medical community, to prepare and minimize the adverse effect on the world. Let's go back to the point that I made at the beginning about Remember now, it was 2001, 2002, of what I called the unholy alliance. The foundation was set up between Dr. Fauci, Bill Gates, and Klaus Schwab of the World Economic Forum, who's the one who's hitting us with supply chain issues right now. And they planned it. They also held an event in 2001 in October of 2019, while the virus is being spread. Yeah, it was called Event 2001, where they had all these people show up preparing for what the world has to do for a pandemic while they were spreading the pandemic. So those are just questions we need to ask ourselves. But in the end, whether this was done purposely for an array of reasons, such as to have an attack on the U.S. by our enemies, Communist China, who, like I said, declared war on the U.S.A. in May of 2019. Right. And this was after the U.S. discovered the internal attacks on the U.S. educational system, U.S. society, and President Trump's administration was confronting them on other issues like trade and militarily. Follow me here, because this is extremely important. Communist China had to remove Trump from power. And Dr. Fauci, along with both the neocons in the Republican Party and the Democrats and financial entities, corporations, and the markets, were knowingly involved. If they were, we're talking about treason here and possible death sentences. Now, with that being said, we have to look at Rand's assessment. I mean, he's might not be looking at the whole big picture, but when you start putting together the big picture, we need concerns. With that, we're going to close part one, and then we'll move to part two, the second round of questioning.